nobody had bothered to ask them, are you dreaming? And I really think it's for the reason that I've been saying periodically during my talk so far. It's because subjective data, we have an aversion to it in science. But it leads us to wrong-headed conclusions to leave out the subjective data, because the fact that dreams and REM sleep correlate does not mean that they're identical. You know, we teach undergraduate students this. Correlation is not causation and all that. So by leaving subjective data out, we can make some dreadful mistakes. It seemed as if REM sleep and dreaming were not the same thing, because damage here leads to a loss of REM sleep with preservation of dreaming. Strengthening this conclusion was the fact that patients with damage to posterior cortex, patients with damage here, these patients had the opposite phenomenology. They had continuation of preservation of REM sleep, but loss of dreams. So with damage in the brain stem, you have loss of REM sleep with preservation of dreams. With damage in this cortical region here, you have loss of dreams with preservation of REM sleep. This is called a double dissociation of function. It showed that they cannot be the same thing. Dreams and REM sleep are two different things. Now, this part of the brain is responsible for, in fact, if any part of the cortex performs the function that Hobson called synthesis, then it's this area, because this is called the inferior parietal lobule. It is where somatosensory information and auditory information and visual information converge. So if you're going to generate a multimodal mental image or hallucination, such as we do in dreams, then no region would be more likely to perform that synthetic function, that bringing together of all of our different perceptual modalities into an experience than this part of the brain. So that wasn't a surprise. What was a surprise, and this is the last of my findings I'm going to mention to you, was that damage in this area had the same consequence. The area that I've marked red. This is the white matter, the white, not the cortex, the white matter, the fibers under the cortex in this part of the brain here, which is called the meso, sort of like the ventromesial quadrant of the frontal lobes, the bottom middle part of the frontal lobes. And unlike this area here, which made good sense that damage here, if you can't generate a mental image, how are you going to generate a dream? The same thing could not be said of this region. It was not by any means clear why damage there should lead to a loss of dreaming. I only had 9 patients, 9 out of my 300, and I think it was 361 cases I ended up with. And only 9 of them had, it's quite hard to get bilateral damage here. So it wasn't surprising that I had such a small number of them, but you can't, 9 is not enough. So the area of damage here are these are facsimiles of the scans in those very 9 patients that I've just mentioned. When I looked at these scans, I was reminded that this area is where we used to do an operation called prefrontal leucotomy, which was with gay abandon. This operation was done in the 30s and 40s of last century for the treatment of psychosis. That is to say, the positive symptoms, hallucinations and delusions were well controlled by this operation. I went back to that literature because in the 1930s and 40s, when psychoanalysis was all the rage, psychiatrists used to talk to their patients about their dreams. And so I thought, well, those psychosurgeons presumably will have observed the same thing that I did in my measly 9 patients. They performed this operation on thousands of patients. So I went back to that old literature, and to my relief, I found that what they reported was the operation had 3 effects. One was the patients no longer had hallucinations and delusions. The second was that they lost spontaneity and motivational drive. And the third was that they stopped dreaming. In fact, it was so widely reported that so generally recognized that these patients lost dreams that one of the scientists concerned, his name was Schindler, observed that if the patient after the operation continues to dream, this is a bad prognostic sign. It means that the operation hasn't worked. And that made a kind of intuitive sense to me, because if you can generate a dream, that means you can generate an hallucination. In fact, dreams are not only hallucinatory, they also are delusional. You believe things which are not true during dreams. And so if you can generate a dream, then presumably you can generate a psychotic symptom. And this led me to the path of answering the question, which was necessarily arose from the findings I've just reported to you, which was, what is it that's going on in this part of the brain that's is so fundamental to the process of dreaming? Clearly damage here, as not only I observed in my little handful of cases, but as had been observed in so many cases in the 1930s and 40s, the damage here leads to a loss of dreaming. But this is spaghetti junction, this part of the brain. There are millions and millions of fiber pathways going through this region. Which one was responsible for generating the dreams? And I hypothesized that it's this one. This is called the mesocortical mesolimbic dopamine circuit. And the reason why I hypothesized that it was this one is because that operation, the prefrontal leucotomy, it was dropped in the 1950s, not for ethical reasons, but because we had discovered a drug. We had discovered antipsychotics, and antipsychotics block dopamine in this pathway. So since this pathway courses through the very region where that cut was made, I thought this was most likely the critical pathway for generating dreams, as it is for generating psychotic symptoms. So from this hypothesis flows predictions. Those predictions are testable, a la Karl Popper. And so, I mean, I knew that cutting through this pathway led to a loss of dreams, but there are many other pathways there too. So it led to the prediction that if you chemically manipulate this pathway, if you increase the level of dopamine activation in this pathway, this should increase the number and frequency, etc., of dreams, and it does. A study done by my colleague Ernest Hartmann showed that if you give patients levodopa, a dopamine booster, they have longer dreams, more frequent dreams, more emotionally intense dreams, more bizarre dreams, more nightmare. Every single dream measure is increased, but not REM sleep. It's something separate. It's the dream that's increased. And my student, Kelvin Kai-Chin Yu, showed that blocking of dopamine by antipsychotics leads to the opposite. Less frequent dreams, shorter dreams, less emotionally intense dreams, etc. So that was one method whereby we tested predictions arising from the hypothesis that this is the circuit that generates dreams. There was another study using a different method. This is positron emission tomography. And this was a study done by my colleague Alan Brown. And he showed, in fact, it was remarkable, the coincidence between his imaging findings and my lesion findings. You will see here the prefrontal cortex, which, as I said, is switched off during sleep, evidently because of the quality of dreams, and my lesion findings showed that damage here has no effect on dreams. Well, his imaging studies showed that this part of the brain is not active during dreaming sleep. Dreaming sleep being REM sleep. Remember, 90% of awakenings from REM sleep, dreams do correlate with REM sleep. So you can operationalize dreaming sleep as REM sleep. And this part of the brain is not activated. But much more interesting, oh, and also this part, the imaging that I said, the synthesis in the parietal cortex, this part is activated. There you have it. But much more interesting is this. This is clearly the main event during dreaming sleep. And this is where that mesocortical, mesolimbic dopamine pathway is located. It is the most active part of the brain during dreaming sleep. So lesions suggest that this is the part that's responsible. Pharmacological studies suggest this is the part that's responsible. Imaging studies reveal the same. And so did these two studies by Dahan and colleagues and Lena and colleagues. What Dahan showed was that he measured with single cell electrodes, he measured the activity in these neurons, the source cells of this circuit, and they fire in in bursts like that, which is the most active that a neuron can be, is when it shows burst firing. So he found prominent burst firing of these neurons during dreaming sleep. And Lena's study measured by a method called microdialysis the release of dopamine at the terminals of this circuit. And the release of dopamine is maximal during dreaming sleep. So all of these different methods, the predictions arising from them, from my hypothesis, were confirmed by all of these methods. And so it seemed like a fairly reasonable, solid conclusion that this was indeed the circuit that drives dreams. I must just mention something about Hobson in passing. Hobson, as you know, had found that cholinergic cells here fire during REM sleep. So on his hypothesis, if you test it in the way I showed you, that a lesion here does not lead to loss of dreams, but a lesion here does. The second of the methods that I mentioned, the pharmacological probes, if you block dopamine, it blocks dreams. What happens if you block acetylcholine? The interesting thing is the very opposite of what Hobson's theory would predict. You block you give anticholinergic drugs, patients have more dreams, not less. In fact, they have dreams while they're awake if you're not careful. So all the evidence pointed to this being the circuit that drives the dream process. So Hobson invited me over to his lab in Harvard to present my findings, and I did so. And he was very, reasonable, decent, accepted the evidence. And at the end of my presentation to his team, I said, Alan, I think you owe Freud an apology, because, if any neuroscientist was to look at this brain and say, how would you expect, if Freud's dream theory was correct, what would you expect to see? Well, Freud says the rational, sort of verbally mediated thinking brain goes to sleep. Well, that's what we see. And it releases from its normal constraints this emotional driven, wishful part of the brain, which is what there's no part of the brain that's more wishful than the mesocortical, mesolimbic dopamine system, as I'll show you in a moment. And there's regression onto the perceptual systems, just as Freud hypothesized. So the dreaming brain looks pretty much like what you would think on the basis of Freudian theory. And the evidence that you adduced against Freud, namely that REM sleep works entirely automatic clockwork switch on off every 90 minutes in a mindless part of the brain, that drives REM sleep, but it doesn't drive dreams. When I went back to London, which is where I was living those days, Hobson sent me an email. In fact, it was the early days of email. And he said, Mark, thanks so much for your presentation. As you know, I find your evidence very compelling, and I'm willing to acknowledge what you found. But did I hear you correctly? At the end, you seem to be saying that these findings support Freud. Because if you're going to interpret your findings as being supportive of Freud, then I'm afraid I'm not willing to acknowledge your findings. So much for objectivity in science. I was absolutely shocked.